In 2015, a team of geneticists at Harvard published a paper in the journal Nature that contained a finding so unexpected that the researchers themselves initially suspected a laboratory error. They were studying the population history of the Americas, building a comprehensive genomic map of indigenous groups across the continent. The work was painstaking, the datasets were large, and the results had been accumulating in the expected pattern, Siberian ancestry predominating across the Americas. The founding Beringian population's genetic signal consistent and recognizable from Alaska to Tierra del Fuego. And then, in a cluster of Amazonian populations, the signal broke. The Surui and Caritiana peoples of the Brazilian Amazon showed a genetic component that matched nothing in the reference panel of Siberian, East Asian, or other Asian populations typically associated with the founding of the Americas. It matched something else entirely. When the researchers ran the analysis against a broader reference panel, the anomalous component showed its closest affinities with populations from Australia, New Guinea, and the Andaman Islands. The same ancient genetic signature that the Denisovan research had connected to the deep human history of the Pacific and Southeast Asia was appearing, in a small but statistically significant proportion, in the genomes of people living in the Amazon rainforest of South America. The researchers checked their methods. They reran the analysis. The signal persisted. They published, carefully, using the term population Y to describe the anomalous ancestry component, after the Tupi word for person, a deliberate choice that avoided naming the signal after any specific geographic origin before its source was understood. And then they asked the question that the data had made unavoidable. How did a genetic signal associated with the ancient populations of the Pacific Rim end up in the Amazon basin? The finding was not, as it turned out, entirely without precedent. Earlier studies had noted anomalous genetic signals in certain South American populations that did not fit the standard Beringian model. The Botocudo people of eastern Brazil, a now largely extinct community, had shown mitochondrial haplogroups that were not found in any other known Native American population. Certain morphological features of the oldest South American skeletal remains, particularly the famous Luzia skull found in Brazil and dated to approximately 11,500 years ago, had been described by some physical anthropologists as resembling the morphology of Australian Aboriginal and Sub-Saharan African populations, more than typical East Asian or Native American populations. These earlier observations had been mostly set aside, either because the morphological comparisons were considered unreliable, or because the mitochondrial data from the Botocudo was too limited to be definitive. The 2015 Harvard study was different. It used whole genome data from living individuals, controlled against a reference panel of hundreds of populations, and produced a statistically robust signal that could not be explained as an artifact of the methodology or as contamination from post-Columbian contact. Population Y was real. The question was what it meant. 3 interpretations have been seriously proposed in the years since the 2015 paper, and the debate between them is still active. The first interpretation proposes that population Y ancestry entered the Americas through Beringia, as part of the initial peopling of the continent, but derived from a founding population that was itself more genetically diverse than the standard Siberian source model assumes. Under this view, the people who crossed from Siberia into Alaska during the last ice age were not a genetically homogeneous group drawn from a single Siberian population. They included individuals or lineages with ancestry connected to the ancient populations of Southeast Asia and the Pacific Rim, ancestry that is now recognized as population Y. As the founding population spread through the Americas and diversified, population Y ancestry became concentrated in some lineages and lost in others, eventually surviving at detectable levels only in certain Amazonian groups. This interpretation has the advantage of requiring no dramatic new migration routes. It explains population Y as a feature of the founding population rather than as a subsequent arrival. The difficulty is that the population Y signal in the Amazonian groups is sufficiently distinct from the standard Beringian ancestry that the modeling required to make it fit a single founding event is complicated. The ancestral population that carried population Y into the Americas would have had to have been genetically very different from the populations that most researchers associate with the Beringian crossing. The second interpretation proposes a coastal Pacific migration. Under this model, Maritime peoples from Southeast Asia or related Pacific populations traveled eastward along a coastal route, possibly along the southern Pacific, making contact with South American coastal populations at some point before the continent was fully settled by populations coming from Beringia. This interpretation connects to the Kelp Highway model that has been proposed for the initial peopling of the Americas and extends it to include a separate, possibly later, wave of Pacific origin migrants reaching South America by sea. The third interpretation, the most dramatic and the most contested, proposes direct transoceanic contact between South America and Australia, or New Guinea, either from east to west or west to east, at a period deep enough in prehistory to have left a detectable genomic signal. The geographic distances involved make this seem almost impossible by the standards of what we know about ancient maritime technology. But the same objection applies to the Polynesian expansion into the remote Pacific, which did happen, and which involved maritime feats of comparable difficulty. The 2015 paper explicitly declined to identify which of these mechanisms was most likely responsible for the population Y signal. The data established the presence of the ancestry. It did not establish how it got there, and subsequent research, including a major 2021 ancient DNA study of South American populations published in Science, confirmed the population Y signal and extended the geographic and temporal picture without definitively resolving the mechanistic question. What the 2021 study did add was temporal resolution. By analyzing ancient DNA from South American individuals spanning thousands of years of pre-Columbian history, the researchers were able to show that population Y ancestry appears in ancient South American genomes dating to several thousand years before the present, and that it is not a recent addition to the Amazon gene pool. Whatever brought it there arrived early, and the Amazonian populations that carry it today are not simply the result of some recent or obscure colonial-era contact. Population Y is ancient in South America. The study also showed that population Y ancestry is not uniform across the Amazon basin. It is concentrated in specific groups, particularly in certain communities of the Brazilian Amazon, and is absent or very low in others. This geographic and ethnic specificity is consistent with a founding event that affected a particular population or set of populations rather than a broad demographic wave that would have spread population Y more evenly across the continent. Consider what this finding means for the story of the first Americans. The Standard Model places a single founding population, derived from Siberian ancestors, as the source of all Native American ancestry. This model is not wrong in its broad outlines. The Beringian crossing is real. The Siberian ancestry is dominant in virtually every tested Native American population, and the demographic history of the Americas is substantially as the model describes. But the model is incomplete. The Americas were not settled by a single homogeneous founding population that then diversified in place. They were settled by a process more complex than any single event model captures, and population Y is the evidence that something in that process has not yet been fully accounted for. The Amazonian peoples who carry population Y ancestry are, in this sense, living archives of a chapter in human prehistory that we do not yet know how to fully read. Their genomes contain information about migrations and contacts and population movements that left no other recoverable record. No tools, no monuments, no written language, just a genetic signal, present in the cells of people who have been living in the Amazon basin for thousands of years, that points toward the Pacific world with a specificity that no model yet fully explains. This is the situation that the ancient DNA revolution keeps producing. Not answers that close questions, but answers that open them, that reveal the inadequacy of the models we have been using and force the construction of new ones that are more complex, more specific, and more honest about the limits of what we know. There is one more dimension to the Amazonian genetic story that deserves particular attention, and it concerns not the population Y signal, but the diversity of the Amazonian populations themselves. The indigenous peoples of the Amazon basin are, as a group, among the most genetically diverse human populations in the Americas. The hundreds of distinct peoples of the Amazon, speaking hundreds of distinct languages in dozens of language families, represent a depth of human diversity that is disproportionate to their numbers. Amazonian language diversity is so extraordinary that the region has been called a linguistic area of maximum diversity, a place where more distinct lineages of human communication have been preserved than anywhere else in the hemisphere. The genetic diversity of Amazonian populations mirrors this linguistic diversity. Different communities show distinct genetic profiles, the product of thousands of years of relative isolation, small population sizes, and the founder effects that come with the establishment of new communities in a vast rainforest. This diversity is itself evidence of the deep time of Amazonian human prehistory. People have been in the Amazon for a very long time, and the genetic distances between contemporary communities reflect the accumulation of that time in the molecules of living people. The population Y signal sits within this context of broader Amazonian diversity. It is not simply an anomaly embedded in an otherwise uniform gene pool. It is one of many unexpected features of a genetic landscape that is far more complex than the simple Beringia model predicts. The Amazon was not settled in a single event and then left alone. It was settled by people who arrived from different directions at different times, who diversified in place over thousands of years, and who maintained such effective isolation from each other that they accumulated genetic distinctiveness at a rate that produces the extraordinary diversity visible in contemporary indigenous Amazonian communities. The question of what happens to this diversity is not merely academic. Many of the Amazonian indigenous communities that carry population Y ancestry, along with dozens of other communities of extraordinary genetic and cultural distinctiveness, are under severe existential pressure from deforestation, land invasion, disease, and the political violence that accompanies the encroachment of the Brazilian agricultural frontier into traditionally indigenous territories. Communities that have maintained their genetic and cultural distinctiveness for thousands of years are being destroyed within decades. The genomic archives that they represent, the records of human prehistory embedded in their cells, are being lost along with the communities themselves. This is not simply a human rights issue, though it is that. It is a scientific catastrophe of the first order. Every community that disappears takes with it genetic information that cannot be reconstructed from the archaeological record alone. The population Y signal was detected because the Sarui and Caritiana communities still exist. If those communities had been destroyed before genomic sequencing was possible, the signal would have been lost with them. The chapter of human prehistory it records closed permanently, without ever having been opened. The Amazonian peoples were never what we thought. We thought, when we thought about them at all, that they were simply the southernmost expression of the Beringian founding population, people who had walked down from Siberia and found their way to the rainforest. The DNA shows something more. A population history of extraordinary complexity, including a signal of ancestry from the other side of the world that no model has fully explained, embedded in communities of such genetic and cultural diversity that their preservation is not just a moral imperative, but a scientific one. The Amazon holds answers to questions about the human past that we have not yet learned how to ask properly. And the communities who carry those answers are still here, still living in the forest that has been their home for thousands of years, waiting for the world to understand what they have always known, that the forest contains more history than any record written outside it has ever managed to capture.